Good afternoon, everyone. I hope that y'all are having a good Friday. And I hope that you've been able to go through the content that I've posted this week. Um, and I hope that if you have any questions, you've either been able to answer them or, oh gosh. I'm sorry, but I'm recording this. Please don't be driving. Uh, I'm sorry to kind of pick on anyone. It's not what I'm intending to do, but I just don't think that's a safe decision. I'm recording it. I'm going to post it on YouTube like I always do, so you can
You're
catch
all
up.
right.
Um, so catalytic mechanisms. That's what we're going to be
Come
going over.
on, go.
Hello? Does someone have? Okay. So we're going to be talking about catalytic mechanisms. If you remember the most recently posted lecture, we were talking about and looking at like free energy diagrams. So we're kind of thinking about this from a kinetic standpoint. But now we're going to switch gears a little bit and we're going to go back to what you probably think about, or personally, whenever I hear the phrase mechanism, I always think of organic chemistry too. And I think of, here's some electrons, we push them over here, and here is a positively charged group that's going to basically accept those electrons. That's what we're going to be talking about. So if you have any questions at any point, please don't hesitate to ask. If there's older terminology from organic chemistry or something of that nature, you need me to, what's the word? This is the word that I like, reconcile it, just let me know. So enzyme reactions are described in the same way as non-catalyzed reactions. For example, we might have an amine that has a lone pair of electrons that is basically targeting a group that has a partial positive charge, like an aldehyde or a ketone. This carbonyl carbon, if you look at it, it's going to have a formal charge or an overall charge of 0, but there's going to be a partial charge because this oxygen right here will have a partial negative charge. So that means that this is going to have a partial positive charge. That's going to lead to the formation of a carbinolamine or carbinolamine intermediate, which leads to an imine or a Schiff base that could form. Now, what I really want you to take away from this is just that we're pushing electrons around. We're finding a group that's electron deficient, and we're moving electrons to that group and targeting it. Now, there are three, this is what's great to me about biochemistry, is that there's three basic types of enzyme mechanisms. There is acid-base catalysis, covalent catalysis, and metal ion catalysis. And I like this because those three terms are pretty, to me, they're self-explanatory in the sense that whenever I hear acid-base, I think of a proton being moved, either a group losing or gaining a proton. An acid is going to be a group that donates a proton. A base would be one that accepts a proton. And so in acid-base catalysis, I think it's pretty reasonable to predict that you're going to have a group that has a proton and the group that accepts a proton. Covalent catalysis is a little bit more nuanced to that than that. You're going to be forming a covalent bond, but the significance is that what are the groups that form that covalent bond? That's going to be the important part. Metal ion catalysis, it's pretty reasonable also to predict that you would have a metal ion. Now, one thing that's worth noting is this Mg2 plus is a metal ion. It has a positive charge. This right here is not a metal ion. It does not have a charge. That's elemental magnesium, not an ion. So that would be an atom. And this is kind of going back to like Gen Chem 1 terminology where you talked about an ion versus an atom. Now, let's get after acid-base catalysis. We're going to introduce these different types of mechanisms, and then we're going to look at some examples of each of them. In acid-base catalysis, this is involved in all enzymatic reactions involving proton transfer. Now, there are a couple of different angles that you can look at this. One is general acid catalysis. A general acid donates a proton to a substrate. And that term general acid, in a biological context, we're thinking about that as more than likely being an amino acid donates a proton to a substrate. Now, there are other situations. For instance, you could have water donate a proton. You could also have a coenzyme donate a proton. Now, in general acid catalysis, this is your kind of what you should think about and what you should hang your hat on with respect to like what's my criteria. We have an active site residue that has to be protonated. So there's an enzyme active site that must be protonated. Now, in general base catalysis, a general base accepts a proton from a substrate. The active site residue must be deprotonated. Now, there are a couple of things to think about with respect to both of these. Now, an active site residue. So that means within, here's my structure of a protein, where I just do an X, that's an amino acid. And that amino acid in general acid catalysis is going to be something like N, H, 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 and that's got a positive charge there. And I'll go ahead and just draw it on back to that. So this is an example of an amino acid that is protonated. By contrast, if we have something like this, N, H, H, with a lone pair of electrons on that nitrogen, that would be an example of a group that's deprotonated. Now, the significance is that this group right here and right here, they have to be able to participate in proton exchange. And so when we're thinking about candidates for this, there's a handful of amino acids that I think are your highest priority, the residues that are most likely to be involved in this, and then there's the residues that are least likely to be involved in this. Your most likely residues are going to be, well, your polar, positive, or negative. And that is true for general acid and general base catalysis. So an amino acid like arginine, for instance, or aspartic acid, those two amino acids can be protonated or deprotonated. Ultimately, their protonation state is influenced by pH of the solution that they're in, but they can go from being positively charged with a proton and negatively charged without a proton, or neutral without a proton. And the same thing down here, R and D, are just two examples. Are those the only amino acids that can participate in this? They are not, but those are some of your most likely. Now, at the same time, there are a handful of amino acids that you can immediately dismiss as being basically incapable of participating in general acid or general base catalysis. Now, does anyone have any idea of amino acids that are likely candidates to, or sorry, least likely candidates to participate in acid-base catalysis? Now, some good examples would be something like glycine or alanine or isoleucine or leucine. These are amino acids that are your nonpolar residues. These nonpolar residues, NP, and then I'm going to go down here and write NP. Your nonpolar residues, well, they can't really lose a proton. So they're not going to be protonated. They're not going to be deprotonated. Sorry. Okay. So then there's a catalytic mechanism, or there's a type of mechanism where you have both general acid and general base, or a general acid and a general base. And an example of this would, or sorry, this is referred to as concerted acid-base catalysis. Now, these are your amino acids that are most likely to participate in acid-base catalysis. glutamic acid, aspartic acid, lysine, arginine, cysteine, histidine, serine, tyrosine. These amino acids are your most likely candidates. Your least likelies are going to be your nonpolar residues. So this is a good list to be familiar with. And it's also super important to be able to either visualize them or draw them in a protonated or a deprotonated state. Now, I want to go ahead and jump ahead for just one moment and look at an example of acid-base catalysis. The enzyme, and just for the purposes of our illustration, I'm going to block out a lot of this. Okay. And then what I'm going to do is I'm going to highlight a specific portion of this. So I'm starting right here in this phosphate group. Then I've got my 5' carbon, 4', 3', 2', 1' nitrogenous base. Then I've got a phosphodiester linkage between my 3' and 5' of my two different sugars. There we go. Okay. And the enzyme that catalyzes this, or the enzyme here, is ribonuclease A. Anytime you see that ribonuclease or deoxyribonuclease, you're talking about a molecule that basically breaks this compound, or you're talking about an enzyme that breaks up that particular molecule. Ribonuclease A is an enzyme that takes a RNA molecule and chops it up. So here's our RNA molecule at the center. See this little semicircle type of thing right here? That right there. That's just a visual, or that's just it's as if someone took the entire protein and they said, let's look at just this one small fraction. Let's look at just the active site. They do that and they say, oh, well, everything in red, that composes your active site of your protein. Sure, all the other 120 some odd amino acids are somewhere else, but we don't really care about all of those. What we are trying to look at is we're distilling this down and saying, we want to look at our substrate, which is highlighted as this yellow molecule in the middle. And then we want to look at the amino acids that are significant for this enzymatic reaction. We've got a pair of amino acids, histidine 119 and histidine number 12. Those are the two amino acids that are important for this catalytic reaction or this catalytic mechanism. Now, what I want you to notice about those amino acids is I'm going to scribble out the arrows. Let's disregard the arrows. His 12 versus his 119. Now, I'd like one person to either add to the chat or speak up and volunteer. What do you notice about the structures of those two amino acids that make them different from one another? And we're thinking about this in the context of acid-base catalysis. Can anyone notice anything about those two amino acids? Okay, we got a chat incoming. We got two chats. Wonderful. His 119. Well, that's positively charged. Okay. His 12 is not positively charged. Why would histidine be positively charged? It's because it's protonated. So effectively, his 119, you could say that this is protonated. And then if you look at his 12 by comparison, it's not. So does anyone object to me referring to his 12 as being deprotonated. Okay. So we've got His 12 and His 119. His 12 is deprotonated. His 119 is protonated. Now, I'm going to erase some of my arrows that I just scribbled in. Histidine 12, I just erased it, and it as a deprotonated residue is running over to the 2' hydroxide group that I'm going to put the 2' hydroxide group, and it's saying, well, I need that proton. So is histidine 12 acting as a base by accepting a proton, or is it or accepting or taking, or is it acting as an acid and donating a proton? Our His 12 as a deprotonated residue is picking up that proton. So it is getting H from 2' OH. So histidine 12 is here acting as a base. Now, I'm going to go ahead and do some more erasing. And well, if histidine 12 is acting as a base and it is deprotonated, then if you look at the arrows for histidine 119, does anyone have a thought as to what histidine 119 is acting as when you compare the structures of the two amino acids? Perfect. It's an acid. So when we think back and we look back to these definitions, I'm going to scroll back super fast. Yeah, these definitions right here. Histidine 119 acts as an acid. It donates a proton to the substrate. His 12 acts as a base by accepting a proton from the substrate. So we have two amino acids in the same active site. One's acting as an acid and one's acting as a base. And this mechanism that we were just looking at is an example of concerted acid-base catalysis, where we have a general acid and a general base participating in our mechanism. Now I'm going to run back to that slide because what we want to do is look at, all right, well, where do we go from here? So if you look at these two or if you look at the lone pair of electrons on histidine 12 picking up that proton, the electrons between that oxygen and hydrogen, which I'm going to highlight in red, those electrons, well, they swing down to our phosphorus. These electrons here swing down to this hydrogen to pick that hydrogen up. And effectively, what we're going to be doing is we're going to be breaking a bond right here between phosphorus and oxygen. And what that is going to do is that's going to lead to this part of our molecule breaking off. So if we think about this RNA molecule, we're severing a bond right there. So we are severing that bond with these two amino acids. We're leading to the breakage of that phosphodiester linkage. And in the process, we're going to release this, which is the same as this. Now, as an intermediate, we're going to have this 2', 3' cyclic nucleotide. Now, one thing I want you to recognize about this is, does anyone see a bond of any sort that connects one of our histidines to our substrate? And let me go ahead and clear almost all of my writing. But look at these first two kind of segments, the top two images. Does anyone see a bond between our histidines and our sugar or our substrate? And I don't hear anything because there isn't one. In this type of mechanism, concerted acid-base catalysis, we don't have a bond between our histidine residues or the enzyme and our substrate. Do we exchange protons with the substrate? 100%. Are these, um, is your enzyme in close proximity to your substrate? 1,000%. Yes, no doubt about it. But we don't have a link or we don't have a direct bond between our histidines and our substrate. Now, everything that is displayed in our right two figures, these right here, well, we've got a water molecule that comes into play. And what's that water molecule going to do? It's going to help us basically break this ring right here, this 2' 3' cyclic nucleotide. We're not going to have a cyclic nucleotide release from this enzyme. And then what we're also going to do is we're going to return histidine 119 to be protonated and histidine number 12 to be deprotonated. So we're essentially using water to regenerate what we previously had. Now, the point of this is that we're trying to break this phosphodiester linkage. We're going to use two histidines, one acting as an acid, one acting as a base, and water. We're going to break that phosphodiester linkage, and we're going to release an RNA molecule that's a little bit shorter. Now, does anyone have any questions about this figure right here in this mechanism? Okay. Well, one of the questions that I always like to ask and get y'all to kind of ponder is, and there's a pretty, what I think is a pretty straightforward answer. Let's say that this histidine, and let's say that this histidine, if these two histidine residues were mutated to another amino acid, and you can go in, goodness, you can go in any direction with this. You could say, oh, I mutate histidine 12 to something else and then 119 to something else. Does anyone have a proposed alternative amino acid that you can mutate these histidines to, and this sort of acid-base catalysis will still take place? Feel free to add to the chat or speak up on this one. Mutating these histidines to another amino acid, what would be a good amino acid to mutate it to? What if I mutated these histidines to a leucine? Thumbs up or thumbs down? Be pretty thumbs down. What about let's go with an aspartic acid. Does aspartic acid side chain, does that R group, does that participate in, um, basically, is that a residue that gets ionized? Absolutely. So we could go to an aspartic acid. What about an arginine? Yeah, pretty reasonable because that could also be an acid or a base. Now, what I want you to think about is the amino acids that you could mutate it to and get the same or potentially have the same result, the amino acids that you could mutate to and basically get no similar result. Your polar residues are your most likely candidates. Your polar positive, polar negative residues are your most likely candidates. Your non-polar residues are your weakest candidates. Okay, so I'm going to go back from this is our example of acid-base catalysis. I have a video where I dissect on my YouTube channel where I dissect this specific amino acid, or sorry, this specific mechanism, and I'll share that link with y'all later today. But this is a really great model for illustrating acid-base catalysis. So now what I'm going to do is I'm going to go back That's our first of our three types of acid-base or covalent. Sorry, just one second. Um, of our different methods of enzyme mechanism. So these are all, or this list right here is also another list of amino acids that you can mutate those histidines to and still potentially get the same result. I would anticipate a question about mutations like that. Mutate this amino acid to what? Do you get the same result? Yes or no? Okay, now we're going to move on to talk about pH profiles. Because if you can imagine, one of the things that's particularly unique about histidine has a pKa of approximately 6.1. It's going to vary a little bit. But if you have a solution with a pH of 1.0, that histidine residue, it's going to be protonated. And so histidine's ability to act as an acid, well, since it's going to be protonated, it's going to be really good. And so that is what you would see right here. Now, if you transition that to a solution with a pH of 13, well, then we're like right over here. What is histidine going to be at a pH of 13? It's going to be deprotonated, so it's not going to act as an acid very well. So when you combine these, when you have concerted acid-base catalysis, you're going to have a graph that looks like this, where you're going to have your amino acid that's acting as a base, where its activity is going to be highest whenever the pH is a little bit higher. But then your amino acids that act as acids, they're going to have a higher activity when the pH is a little bit lower. Now, this x-axis down here, it's just labeled as pH. We don't have it defined because this is going to vary quite a bit. Some enzymes are going to have a pH range where they are most active from 11 to 11.5. Anything above or below 11.5 and 11 respectively, the activity is going to be super low. And that enzyme effectively is going to be inactive. Now, our next type of catalysis, covalent catalysis. In covalent catalysis, a covalent bond is transiently formed between the substrate and the enzyme or the coenzyme. This reaction usually involves a nucleophilic group, a nucleophile, on the enzyme and an electrophile on the substrate. So I always think about that as a negative charged or a negatively charged group on the enzyme and a positively charged group on the substrate. Now, here's an example of an enzyme known as chymotrypsin. Chymotrypsin is a protease. A protease is a protein, or it's a type of enzyme that cuts peptide bonds. So chymotrypsin has a serine residue, serine 195. And somewhere on chymotrypsin, it has an amino acid in close proximity to that serine 195. And what it is able to do is it's able to abstract a proton. And this basic residue is protonated, which makes serine 195 act as a super strong nucleophile. Now, amino acids that are strong candidates to act as nucleophiles, I want you to look at this and be able to think like, okay, what are those residues? Let's look at the bottom here. Which amino acid is a five-membered ring that we just talked about? Histidine. So histidine is a great amino acid for acid-base catalysis and a pretty good one for covalent catalysis. An amino acid with an R group that looks like this, well, arginine and lysine are both good candidates. An amino acid with a sulfhydryl group, cysteine. An amino acid with a hydroxyl group, you got three of them: serine, threonine, and tyrosine. I want you to know those different amino acids as this is a good nucleophile, this is not a good nucleophile. Again, where do these amino acids fall? They fall in the realm of polar residues. And these happen to be polar positive or polar uncharged, but think about them as okay, whenever I'm like, if you're interested or if you are thinking like, oh, which amino acids are most interesting? It's your polar residues. Your non-polar residues, they're a little bit boring, but that sounds very personal of me. Now, electrophiles. Where is an electrophile going to be found? It's going to be found on your substrate. Now, biologically important electrophiles generally are either positively charged or contain an unfilled valence electron shells or contain an electropositive atom. Strong electrophiles are protons, metal ions, carbonyl carbon atoms, and cationic amines or Schiff bases. So we're looking at a carbonyl carbon. Aldehydes and ketones love to do that. Protons and metal ions are also good candidates. But when we talk about metal ions, it's also important that you're not venturing too far into metal ion catalysis, which we will come back to because what I'd like to do now is talk about an example of covalent catalysis. Now, the enzyme that we're going to use for covalent catalysis is an enzyme known as lysozyme. Lysozyme hydrolyzes the peptidoglycan layer of bacterial cell walls by breaking the beta-1,4 glycosidic linkage between NAM and NAG. NAM, NAG. This bond right here is what's broken between them. So that's N-acetamiramate and N-acetylglucosamine, NAM and NAG. So this is which happens at that glycosidic linkage, that beta-1,4 glycosidic linkage. Now, the input looks like this, the output looks like this. Whenever we are looking at this enzyme and what it's doing, there's a couple of things to take away from it. This figure on the right-hand side, we're going to come back to. But what this, actually, we'll tackle it right now. This shows the pattern. So if you look at this thing in the middle, that's NAG, NAM, NAG, NAM. What is it doing that? Minimize. NAM, NAG, NAM. Okay. Now, what we're going to be doing is lysozyme is going to cut between NAM and NAG. Okay. So it's going to be cutting on that will be the anomeric carbon from 1 to 4 of NAM. Whenever lysozyme sees the one, or when it sees the anomeric carbon of NAG, it just ignores it. The amino acids that are critical for this are aspartate 52, glutamate 35, and those are your pretty key amino acids. Now, what's tricky about this is you have a handful of other amino acids. So if you start at the top, you'll see aspartate 101, tryptophan 62, tryptophan 63, asparagine 59, alanine 107. And if you look closely at that alanine, that's not what alanine looks like, but that's what the backbone of alanine looks like. I mean, it's the backbone of every amino acid. But this right here is illustrating that we have a hydrogen bond between our substrate and a amino acid's backbone. Another amino acid involved, valine 109, glutamine 57, glutamine 57 again, asparagine 44, glutamic acid 35, phenylalanine 34, asparagine 37, arginine 114. Okay, so all of these different amino acids are involved in this mechanism. But one of the things I said a second ago was that really at the end of the day, it's these two amino acids that are most critical. What does that mean for everything else? What that means is that all these other amino acids are simply there to kind of properly position our substrate, properly position and basically hold in place this polysaccharide. It's almost like um, when you go to get your hair cut, person who's cutting your hair, they'll hold out your hair and then they'll use the scissors and clip, or they'll use the clippers. But just for this example, we're holding out the hair and we're cutting. Now, if they did not hold your hair out, they don't have a good sense of how long your hair is. They don't have a good sense of how much they need to cut, where they should cut. If you're like, oh, well, cut an inch off my hair, they might be thinking, well, did they mean from the head or did they mean from the end of the hair? Hmm. And if they pull your hair out like that, not pull it, but extend it outward, then they'll have a sense of like, okay, cut an inch off. I'm gonna ask, should I cut 1 inch from your scalp or 1 inch from the end of the hair? All of these amino acids, what they are doing is helping to position our polysaccharide so that we can cut in between the correct sugars in this polysaccharide. The ones that are actually doing it, the scissors that are actually doing this, are aspartate 52 and glutamic acid 35, which we're going to see right here. Here is a proposed mechanism for the cleavage of this polysaccharide. The two amino acids that I mentioned, glutamic acid 35, aspartate 52. If we look at this cell, here, 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 what you'll notice is that there are only two amino acids that are highlighted because the other amino acids are not directly involved in the breaking of this bond. They're essential supporters, but they are not going to make or break a bond. Now, remember, this is covalent catalysis. Glutamic acid 35. Let's look at this residue. Glutamic acid 35. Oh, it's protonated. Oh, snap. It gives up a proton. It acts as an acid. Acts as acid. That's not a mistake. So why is this acid-base or why isn't this acid-base catalysis? Well, we can't answer that question with this right here. But what we can say is glutamic acid starts out this reaction by acting as an acid. It donates a proton. And whenever it donates a proton, here's our general acid catalysis, glutamate 35 is deprotonated. It's got a negative charge. Now, when it donates that, our glycosidic linkage is broken. And so our sugar or our polysaccharide is basically released. Now, we immediately have an intermediate, the oxonium ion. This is a transition state intermediate. And lo and behold, we have a carbocation. That positively charged carbon, what it is going to do is it is going to react with this negatively charged aspartate 52. That aspartate 52 and that positively charged carbon of our ring, we're going to form a bond between our sugar and our aspartate 52. Now, does anyone, and I'll leave this for the chat, would anyone either like to say it aloud or add it to the chat? What type of bond is formed between our aspartate 52 and our sugar? Is it a hydrogen bond? No. Is it a covalent bond? I like it. Yes, it absolutely is. So now let's think about our definitions of acid-base catalysis and covalent catalysis. Acid-base catalysis, you got to have an amino acid that acts as a proton donor. Glutamic acid 35 did that. So if you looked at just the first step, you'd say, oh, well, this is going to follow an acid-base catalyzed mechanism. Then when you get down to the formation of this intermediate down here, you're going to say, well, it forms a covalent bond with the substrate. It's almost like that takes precedence, and that is greater significance. So as soon as we form that covalent bond between our enzyme and our substrate, then all of a sudden, this whole thing, it doesn't matter if there's 50 steps of acid-base catalysis. Doesn't matter if there's 50 acids and 312 bases. As soon as you establish that covalent bond with your substrate, it's covalent catalysis. Was acid-base catalysis involved? Yeah, absolutely. But our kind of definition for this type of reaction, we would look at this as covalent catalysis. So then the important things that you need to know about this are to be able to discern or decipher, well, what's aspartate 52? It's a nucleophile. Our oxonium ion, that is an electrophile. What we're going to do is we want to get glutamic acid 35 back to being a protonated residue, because at this point, it's still deprotonated. We also want aspartate 52 to get back to its good state where it can act as a nucleophile. And so what are we going to have? We're going to say, hello, water. We need you because what you're going to do is you're going to act as an acid, donate a proton up to glutamic acid 35. And then what you're also going to do is you're going to act as a base. And when you act as a base, you're going to allow this aspartate 52 to be released. So water acts as a acid and a base. Glutamic acid acts as an acid, and aspartate 52 acts as a nucleophile. Our electrophile was a substrate. Does anyone have any questions about covalent catalysis? Great. Now, our next type of catalysis is metal ion catalysis. Metal ion catalysis, well, nearly 1/3 of all known enzymes require the presence of metals for catalytic activity. There are enzymes known as metalloenzymes. These contain tightly bound metal cofactors such as iron 2+, 3+, copper 2+, zinc 2+, manganese, and cobalt 2+. So a metalloenzyme is going to have a tightly bound metal, one of these metals. There are also metal activated enzymes, and they only loosely bind the ions. The ions are usually sodium, potassium, magnesium, or calcium. Now, there's a piece of terminology when it comes to metalloenzymes and metal activated enzymes. You might refer to something as the apo form of an enzyme, or this is the apo enzyme. Sorry, that is the inactive form of the enzyme in the absence of the cofactor. The halo form is the active form of the enzyme, and the cofactor is present. Now, an example of a metal ion catalyzed reaction is carbonic anhydrase. Carbonic anhydrase takes CO2 and H2O and makes HCO3- and H+. This is an essential enzyme for trafficking carbon dioxide from your muscle tissues. Now, with that in mind, does anyone know what the pKa of water is? pKa of water is about 14. So the way that this enzyme works, what I'd like to draw your attention to is this first circle that I've looked at right here. I want you to recognize a couple of things. One is that we have the metal ion zinc 2+. That zinc is basically held in place by 1, 2, 3 histidine residues. Now, have we seen another instance of a histidine residue holding a divalent cation in place or helping to hold a divalent cation in place? Spoiler alert, yes, we have. Does anyone know where we've seen a histidine residue help to hold a divalent cation in place? I'm going to give you a hint. And that's not me yawning. What am I doing? I'm bringing a bunch of oxygen into my lungs. Where's that oxygen going to want to go? It's going to want to get to my peripheral tissue. How's it going to get there? What protein? Well, hemoglobin. That would be correct. Yeah. Hemoglobin is going to, or hemoglobin is a protein that has a couple of histidine residues that hold in place an Fe2 plus cation. That Fe2 plus helps hold in place O2. Okay. Now, I just bring that up because I like to kind of set the stage as here's sort of precedence for what we've seen before. Now, what this zinc ion is going to do is this zinc ion is going to hold in place water or H2O. But whenever it does that, that interaction between zinc and water causes the pKa of water to go from 14 to 7. Okay. So this particular water molecule, this particular water molecule right here, if it has a pKa of 14 and it is in a solution with a pH of 7.5, then, basically 0% of H2O is deprotonated. Now, if that pKa drops from 14 down to 7, and the solution's pH drops to, or is at 7.5, that's an instance where our pH is greater than our pKa. So this water molecule, what it is going to do is it's going to lose this proton. When it loses that proton, what's going to happen to the pH of our environment? It's going to go down. That's one thing that's worth noting. Then what is also going to happen is this hydroxide group is going to love to be able to interact with something that has a partial positive charge. And lo and behold, carbon dioxide is accessible and available. So this lone pair of electrons, or one of these lone pairs of electrons on oxygen, what it's going to do is go after this partial positive charge on our carbon. And it is going to form a bond between oxygen and carbon. This negatively charged oxygen is going to be stabilized by zinc 2 plus. And what we're going to release then is HCO3 minus. And that HCO3 minus is going to be displaced by H2O. So effectively what we're able to do is carbonic anhydrase grabs onto a water molecule and more or less converts it almost to a hydroxide ion. That hydroxide ion is ready to react with carbon dioxide. That carbon dioxide, when it reacts with that hydroxide ion, forms HCO3, which makes CO2 so much more soluble, which makes CO2 something that's very capable of perfusing through the bloodstream. So this is what a metal ion, or this is an example of metal ion catalysis. One of the things that's very important to know whenever we jump back to this right here, when we think about metalloenzymes and metal activated enzymes, let's think about metalloenzymes. Contain tightly bound metal cofactors such as iron, copper, zinc, manganese, or cobalt. What amino acids do you think would be really good candidates for holding those different metal cations in place? Well, something that's got a lone pair of electrons. Well, histidine's a fantastic option because at a neutral pH, chances are histidine's got a lone pair of electrons, so it's looking for a positively charged group, like a metal ion. So my takeaway from this, and what I want y'all to take away from this, is that if there's an amino acid that is very dynamic and can participate in acid-base, covalent, and metal ion catalysis, we can histidine, then which can act as a nucleophile, histidine, which can stabilize a metal ion for metal ion catalysis, histidine. Histidine is kind of our most dynamic enzyme when it comes to different types of catalysis. Does anyone have any questions about what we've covered so far, our three major types of catalysis? Okay. Now, what I think are the important items for y'all to be familiar with, the different amino acids, what they do, what they're likely to do, how they will participate in any sort of catalysis, and then thinking about, well, if this residue is mutated to this, will this reaction continue to take place? Those are the big items. Now, with that said, I'm going to go ahead and go through another example, and I'm going to dig a little bit deeper into a type of covalent catalysis, in part because covalent catalysis also has some elements of acid-base catalysis. Now, I mentioned this class of enzymes previously. This is a protease. Proteases are examples of hydrolases, which when you think back to your EC numbering scheme, where were hydrolases? If number one was a oxidoreductase, 5 are isomerases, 6 are liases or ligases, 4 are liases. I think it's 3 that are hydrolases. So proteases fall under that category of hydrolases. Now, proteases are proteins that cut proteins. That sounds so strange, but that's exactly what they are. These are molecules that or that are polymers of amino acids, and they use those amino acids to cut other polymers of amino acids. Now, what they do is they cut a peptide bond. So when you think about drawing a peptide, I always think N, C, C, N, C, C. And I'm going to say this versus N, C, C. If we have a protease, it is going to cut a peptide bond. When we look at option A and option B here, well, A has a peptide bond, and that peptide bond is right here. I'm going to go ahead and box. No, I'm not going to box the whole thing. I'm just going to box the peptide bond. It's this nitrogen and carbon-nitrogen bond that is a peptide bond. So option A has a peptide bond. Now option B, what do you think a protease is going to do when it sees option B? It's going to basically jump on right here, and it's going to scan. It's going to be like, all right, I'm looking for a carbon to nitrogen. Oh, I see nitrogen and carbon, not a peptide bond, don't care. Okay, scanning, scanning, scanning. Now I've reached the end of the molecule. I never found my bond. A protease is going to see a singular amino acid and go, I don't care about this because this does not have the peptide bond that I'm looking for. I feel like there's a Star Wars reference there. I don't know if people are, what's it? Star Trek people are Trekkies. So Star Wars people, I'm going to say, are Trekkies. But these are not the substrates that you're looking for. They completed the Star Wars reference. Okay, a serine protease. Now, serine proteases are a class or a family of proteolytic enzymes that have an active site serine. Chymotrypsin and subtilisin are two examples. Those are two specific enzymes that are serine proteases. Thrombin is another serine protease, and thrombin is crucial in the blood clotting cascade. So this is essential for making sure that you stop bleeding. Uh, Trypsin, chymotrypsin, and elastase are all digestive enzymes, so they're found in your gut or in the guts of other organisms. Subtilisin is a bacterial protease. Plasmin breaks down the fibrin polymers of blood clots. Um, and then if you tissue plasminogen activator or TPA cleaves the proenzyme plasminogen, yielding plasmin. So if you've ever known someone who has a stroke, the medication that they are administered ideally within the first 3 hours of the injury is TPA or tissue plasminogen activator. It's also known as the clot buster. Clot buster, not bluster. Sorry. Clot buster is supposed to break up any sort of blood clots. That's the intent. Uh, and whenever it is administered, it's pretty miraculous. Okay, so now to visualize the action of a protease, we'll look at a class of molecules known as zymogens. Now, this sounds like it's straight off of a movie or something like that, or like a sci-fi movie. Um, Zymogens are it's 256. Um, Zymogens are proteins that basically they float around and they don't do anything. Now, they don't do anything because they are activated by proteolytic cleavage. Some enzymes are initially produced in an inactive form. This is their zymogen form. A portion of the protein chain must be removed in order to activate it. This activation is irreversible. So think about it as like, um, well, this is an enzyme, this chymotrypsinogen. This is in its inactive zymogen form. So that full polypeptide is produced, it's not used. However, there is going to be an instance where there is another protease known as trypsin. Trypsin will be activated. It will go to chymotrypsinogen and it will cleave a peptide bond. And it will produce high chymotrypsin. And here what we have is our full polypeptide. We've got a bunch of disulfide bonds here, 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 and here. This peptide bond that's broken here, well, that polypeptide is going to remain intact. This pi chymotrypsin will then undergo a process known as self-digestion or autoactivation, where it's going to cut a couple of other peptide bonds and release a pair of, it's almost like it says, okay, well, something cut my arm, so I'm going to cut my arm off, and then I'm going to cut my other arm off, and now I'm ready to go. Now that's an extreme example and not realistic because proteins don't have arms. But it's going to be fully active after it activates itself, yielding this alpha chymotrypsin. Now, the reason that I bring this up is because think about what a protease is. It's a protein that cuts peptide bonds. Do you want something that is cutting, just kind of willy-nilly going out there and cutting peptide bonds? No, you don't. Because if there is a enzyme that cuts peptide bonds, that's almost like a self-destruct mechanism. You only want that active under certain circumstances. One example would be a process of programmed cell death or autophagy, or cellular senescence. All of these are kind of cell death cascades where, hey, this cell is being threatened. So what are we going to do? Well, we're going to take all of the resources that we have, all the nutrients that we have, and basically blow them up so we can recycle them. And we're going to use proteases to basically take proteins that are currently out there, chop them up, so then all of those amino acids can go be released in some other form or fashion, or be used in some other fashion. Now, all of these proteases vary in terms of what they cut after. So they are going to look at the primary structure of a protein and they're going to say, well, I'm going to cut after this amino acid. So trypsin, chymotrypsin, and elastase are all proteases that are going to cut with a different pattern. Trypsin cleaves on the C-terminal side of a basic positively charged amino acid. So if we think about a peptide sequence like this, alanine, arginine, alanine, okay? So NCC, NCC, NCC. Okay, so this is my alanine, R sub 1, and then this is my R sub 2, which is my arginine, and then here's my R sub 3, R sub 1, R sub 2, R sub 3. Okay, trypsin is going to cut on the C termini of a positively charged residue. Is alanine a positively charged residue? Nope. So what trypsin's going to do is it's going to look at this first residue and it's going to say, okay, well, that first residue, it's not a positively charged residue, so I don't care. Now I'm going to go on to my next R group. My next R group happens to be an arginine. Okay, great. Now I'm going to cut on the C termini. So cut between, or cut the peptide bond between here and here. It's going to cut that peptide bond and it's going to release this and this as separate products. That's what trypsin is going to do. Chymotrypsin is going to cut on the C termini side or C terminal side of an aromatic amino acid. Elastase is going to cut on the C terminal side of a small neutral amino acid. Now, the way that that's done is with a part of the protein known as a specificity pocket. The way that serine proteases work mechanistically is they have a total of 3 significant amino acids. Every serine protease has these 3 amino acids. It's got a histidine in position 57, a serine in 195, and a aspartate in 102. These are what are known as the active site residues. These active site residues are responsible for actually breaking that peptide bond. We've got serine acting as a nucleophile, histidine as a general base, and aspartate, really, if you've ever seen the movie The Big Lebowski, aspartate really just acts to tie the room together. That's a reference to The Big Lebowski in regards to the central character's rug. Aspartate 102, it really just kind of holds the histidine in the right place. It doesn't do anything mechanistically to like break a bond or anything like that. It's just kind of there and it's like, hey, histidine, just hang out where you are. Don't like do anything wild or crazy. Just cut the bond, okay? Now, what chymotrypsin is going to do is it's going to take a peptide, it's going to take water, and it's going to make 2 peptides. Chymotrypsin catalyzes a rate enhancement of 10 to the 9th. So this is going to be 10 to the 9th times faster than the uncatalyzed reaction. The mechanism involves an acylation and deacylation reaction rather than direct attack by H2O. What we're going to see with this is also a level of regulation by nitrophenol, which I'm not going to ask you anything about this. What I instead want you to take away from this slide are these 2 items right here. Now, the chymotrypsin mechanism. Here is our substrate. At the top of this figure is our substrate. 5, 6, 7. That's our substrate. And at the bottom of this figure are all of the amino acids that are involved in this. So aspartate, histidine, serine. What I think is really effective is to look at each one of these amino acids individually and see how they change. If we track aspartate 102, it's negatively charged across every single one of these. Why is that? Because all that it's doing is it's hydrogen bonding with this histidine. Let's now look at histidine. Well, in the first cell, it's deprotonated and it has no overall charge. In the second cell, well, now it's protonated, so that histidine would have a positive charge. In a third, it's deprotonated, fourth, deprotonated, fifth, deprotonated, sixth, protonated, seventh, deprotonated. So basically, that histidine goes between being protonated and deprotonated. It donates a proton and accepts a proton periodically. It starts out by accepting a proton. Where is it accepting that proton from? Well, it's accepting a proton from the adjacent serine. So it's acting as a base in comparison to that serine. That serine is going to act as a nucleophile to form the covalent bond between the substrate and itself. Now, what that is going to do, and when it comes to enzymatic mechanisms, I want you to be familiar with what each of the amino acids do. Um, is it acting as a base? Is it acting as an acid, a nucleophile, etc.? That's the sort of question that I'm going to ask you, and that's the sort of thing that you should expect on your exam on Monday. Um, I will not ask you a question where I say, hey, recall between steps 6 and 7, what happens? Instead, what you should expect is, hey, I'm going to give you this figure, and or maybe I'll give you these 2 steps. These 2 steps, and I'll say, all right, well, based on what you see between 5 and 6, what did water do with respect to histidine? Well, if you look at that, you'll see that histidine is deprotonated. And then in cell 6, it's protonated. That oxygen, that OH group, went to our carbon there. So it's pretty safe to say that water acted as an acid, donated a proton to histidine, and then later that histidine was able to act as an acid itself by donating that proton. So histidine accepts a proton from water, then donates that same proton. Those are the sorts of questions that that you should expect from me. So histidine basically acts as a general base. Serine is our strong nucleophile. Now, these next 2 slides are the exact same figures, just extrapolated outward, and some of them are copied. So this is just, if you want to look at them on a larger scale, that's what this is for. Now, protease inhibitors. All of the new drugs on the market to treat HIV and AIDS are protease inhibitors, and they're specific inhibitors of HIV-1 protease. These drugs are developed through a structure-based design strategy. So if you think back to that structure-based design, or that idea of structure-based design, think back to secondary structure and the work of Chow and Fassman. They said, hey, use the primary structure and you can predict a secondary structure. Okay, cool. The way that in the direction that research is going is, can we use the primary structure to predict a tertiary structure? And if you're familiar at all with AlphaFold, which is a project of Google's, well, Alphabet being the parent company of Google, I always feel hokey saying that, but what they do, or one of the projects that they've done, is they've said, can we solve a protein structure using just the primary structure? If we can, then we can take just this string of amino acids and say, this is what the protein folds like in 3D space. And the sooner that you know that, and the more confident and certain that you are, then you can say, all right, well, here's our protein, and here's this little pocket right there. You know what? I bet we could take sequinavir right here, and let's methylate it right here, and then let's add a phosphate group right here. And I think that we're going to make a drug that is more potent, because these negative charges right here will be attracted to positive charges here. So that's going to be held in place more. Oh, this is perfect. And that's how protease inhibitors are designed. Looking at a protease and saying, all right, well, how does this protease function? The better that we know that, the better off we are in terms of basically fighting it off. Okay, now I'm going to skip that. Now what I want to go on to is the last thing, and that is enzyme regulation. So one way that some enzymes can be regulated, or a protease can be regulated, is proteases are regulated by activation by proteolytic cleavage. Well, if you can prevent that, that's the way that a protease is going to be regulated. But other enzymes are regulated in a number of different ways. There are enzymes that have similar but not identical amino acid sequences. Um, for example, hexokinase and glucokinase, which are highlighted down here. Hexokinase is ubiquitous. It's in the muscle or in your muscles. Glucokinase, by comparison, is found basically just in the liver. And these two enzymes differ in that hexokinase, found in the muscle, will take any 6-carbon sugar. We'll take glucose, fructose, mannose, allose. Glucokinase, by contrast, only wants glucose. If you give glucokinase some mannose, it says, I'll work with this if this is the only sugar that's available. But I really don't want to work with it. Now, another way that enzymes can be regulated is through they can be allosterically regulated. Now, we saw and we talked about allostery in the context of hemoglobin. Hemoglobin has two different forms, the T state and the R state. Hemoglobin in the T state is the deoxy form of hemoglobin. The R state is the oxygenated form of hemoglobin. There are typically an allosteric enzyme has multiple subunits, a catalytic and a regulatory subunit. So what that means is that the catalytic subunit is the part that's the business end, whereas the regulatory subunit is more or less the subunit that basically controls the catalytic subunit. So our C and our R here. These two parts of the protein have different functions, as I've mentioned. They have different affinities. The substrate will bind to the catalytic domain or catalytic portion, whereas an activator molecule will bind to the regulatory domain. So this R is going to bind to a molecule, a small molecule that is basically a positive modulator. So it's kind of going to be the light switch that turns on by changing the shape of the protein, it turns on the catalytic domain. So we see this positive modulator bind to our regulatory domain, which causes this site where our substrate naturally wants to bind, the shape of it changes. So now all of a sudden, if you look at the shape of our substrate, a triangle, it's not going to fit in here. However, whenever the positive modulator binds to our whole protein and causes the shape of the protein to change, now all of a sudden our substrate has a binding site that's much more favorable to bind to. And that's going to give us our active enzyme substrate complex. This is our catalytic and our regulatory domain, where both of the ideal binding partners are formed. Now, similarly, there are negative modulators. There are inhibitors. For example, here what we see is the metabolic pathway going from threonine, the amino acid threonine, being converted to isoleucine. Now, isoleucine serves as a feedback inhibitor by inhibiting the function of threonine dehydratase. When isoleucine binds to threonine dehydratase, well, then the activity of threonine dehydratase plummets. It's a feedback loop in which isoleucine basically says, hey, there's a high concentration of me. We need to stop making me. And where I'm going to attack this, I'm going straight to the top. I'm saying threonine dehydratase, stop acting, stop working, because there's too much of me present. Now, this next, I've got two left. I'm going to skip over this because we have not talked about Vmax and Km, so don't worry about those. We'll talk about those next week. Covalent modification is or there's a couple of examples of modification. So when a protein gets produced, so here's our protein folded in 3D space. And somewhere on this protein is a tyrosine residue. Here's our tyrosine residue. Well, there is a process known as adenylation where a molecule of ATP will be taken by an enzyme and basically AMP will take the place of this hydrogen. So this whole thing right here will be added right here where I just put my star. Now, this addition is reversible. I want you to think about this in a couple of different ways. One way that I want you to think about this is from a molecular standpoint. And I'm going to ask a question. Is OH, just that functional group, is a hydroxyl group, larger than, let's go ahead and say, the entire AMP? Is OH, that functional group, larger than an AMP molecule? Go ahead and add to the chat or feel free to speak up on this one. A hydroxide group is not larger than AMP. So the space that the hydroxide group occupied now is occupied by AMP. That's a massive molecule by comparison. That hydroxide group has been replaced by this whole thing right here. Now, with that in mind, do you think that the interactions that AMP has are going to be different than hydroxyl group? Totally. For instance, a hydroxyl group, what type of interactions can it have? Well, it could do some hydrogen bonding. Yeah, totally. True. Okay. If you think about one OH group, well, chances are it's going to have one H bond. Cool. That's great. Now, if we think about adenine, okay, well, do we know that adenine hydrogen bonds with anything? Yeah, we do. We have on pretty good authority that adenine likes to hydrogen bond with, well, uracil or thymine. So it likes to have two H bonds, for starters. In addition to that, here's a hydroxide group. Here's a hydroxide group. So at very least, we're going from one hydrogen bond to four hydrogen bonds. So the way that I want you to think about this is when you replace a hydroxide group with something else, like an adenylyl group, the types of interactions that it can participate in or with have changed as well. Now, what that's going to mean is the shape of the protein can change. And there's a strong relationship between the shape of a protein and the function of a protein. So in many cases, adding a group like AMP might activate an enzyme, or it might inhibit its activity. So think about any sort of post-translational modification like this, a covalent modification, as well, it's changing the interactions on the molecular or the atomic level, but it's also, or by virtue of that, it's kind of acting as a light switch, activating or deactivating the enzyme. Now, next covalent modification that I'd like to talk about is a methylation. Methylations are typically added to glutamic acid residues, which, if it wasn't clear enough, the targets for adenylation are tyrosine. Good test question. The targets for methylation, glutamic acid. Good test question. If you think about a glutamic acid, what types of interactions will the glutamate like to do? Well, glutamic acid likes ionic interactions. If you add a methyl group on there, it's not going to be good for ionic interactions. It's going to be good for hydrophobic interactions, if anything. Now, our last example of a covalent modification is phosphorylation. There are 4 different amino acids that phosphate groups like to get added to. And what do you notice about three of these four? They all have hydroxyl groups, tyrosine, serine, threonine, all like to have that OH replaced by O, P, O, O. There we go. Now, that phosphate group, so we've got hydrogen bonding versus ionic interactions. Now, what this is showing down here, sorry, on the right-hand side, is an enzyme known as phosphorylase B. Phosphorylase B is a dimer. Phosphorylase B has two serine residues on each one of its monomeric subunits. So serine number 14 on each one of those polypeptides gets a phosphate group added to it. And whenever that happens, when phosphorylase B is phosphorylated, it is now converted from the B form, the less active form of the enzyme, to the A form, the more active form of the enzyme. So we have phosphorylase A, our active form of our enzyme. Now, the enzyme that adds those phosphate groups is phosphorylase kinase. Phosphorylase phosphatase does the reciprocal action by removing those phosphates and releasing those phosphates as inorganic phosphate. So what I want you to think about is, and be able to answer with respect to questions pertaining to this, is the addition of a phosphate is more than likely going to activate a protein. That is going to be done by a transferase, specifically a kinase. Removal of phosphate groups from a protein, that's going to be done by a phosphatase. That phosphatase will remove those phosphates as inorganic phosphates. And releasing those will deactivate that protein. Okay, so we've talked about quite a bit of stuff today. As an overview, I want you to be familiar with different amino acids, what roles they play in different types of catalysis. I want you to be familiar with types of modifications that different amino acids can undergo. And how does that lead to, or the terminology associated with this. So adenylation, what is that? Well, that's an addition of an AMP. Okay, well, that's essentially what I wanted to cover today. I'm going to go ahead and stop my recording. If you have any questions, you're welcome to hang around.